still fluctuating because there are so many things like a list of people.
All right, I'm gonna pass back your exams. Like I said, last week I did add 6% to each of your scores at the curve. I'm sorry. Using it.
I was there. I was just like,
That's
What was the original pure one?
yeah, the 66.
Okay.
Anybody not get their exam? All right. All right, let's go ahead and get started. So today we're going to continue on some microbial ecology from lecture 11, and then I'm going to introduce a little bit of some culture-independent microbiology techniques and what that means. And so we ended off over here last week. So we were talking about different species in a community and how they can affect that community if they're lost. And so we stopped at talking about keystone species. And so these are species that plays a role in its community. It doesn't really have to exist in really large numbers. Its relative abundance doesn't necessarily have to be really high, but it plays a really important role that if it is lost from the community, it's going to have a disproportionately large effect on the entire community as a whole. And so, like I said, typically it's not the most abundant species or the major energy producer, but it's vital to the maintenance of that community. So keystone species typically exist in low numbers, and so they are really important and sources of something that's really important for that community. So we found that the same ecological concepts that govern the macro scale relationships seem to apply to microbial communities inside of us as well. So this is an example of something we call ecological succession. And so if you look up here at the top, so if you have a community or an ecosystem, and that ecosystem, that forest gets set on fire, that forest ecosystem has to recover from the fire. And so while it's recovering, so this is the damaged state, and then over time the recovered state. So first you have this forest fire, and so once you have that damage, you'll start to have some pioneer species like grasses that will move into that burned area at first, and they'll grow, and them being able to grow then paves the way for intermediate species to then come in, like shrubs and other smaller trees. And then ultimately, once those grow, then all the other species are able to return. So that's those trees in the forest. So this is what we call ecological succession, where there's damage to community and then it can grow back to the way that it was over time. And so the key here is that you need these primary or pioneer species to colonize that area and in order for the intermediate species to grow and then the climatic species to grow as well. And so studies have described a similar process in the gut microbiome following antibiotic therapy. And so basically if you have antibiotics, so right here I'm showing you the intestinal epithelial cells. If you're given antibiotics, that's going to kill the majority of the microbes there. And so following antibiotic treatment and removal of the antibiotic, you'll start to have these primary species growing in that community. And so these pave the way for secondary species to come back and grow. And then those also then pave the way for these tertiary species to colonize that area. And so basically these bacteria, the blue and the purple, require this recovering ecosystem in order for them to survive and to grow. And so just to give you a little bit context here as well, this red shading is the mucus layer. So when you have disruption to this intestinal barrier by antibiotics, for example, you get thinning of this mucus layer. And then that's actually one of the studies that I'm doing for my dissertation, and that's one of the things I'm seeing. So good to see that my work is being reiterated. But yeah, so it's really important to have that mucus layer come back up so that these microbes can colonize as well. So there's so many different things at play here. Does that make sense? So like I said, this is called ecological succession. And so this is a predictable process because we know that, over time, the changes in the species composition of communities will start to reestablish themselves, and it's kind of, a predictable process in terms of some primary species that need to be there in order for secondary and tertiary species to grow. And so ecological succession can occur either in an environment that are devoid of any pre-existing communities, so there's nothing there, or in environments where there's been a massive community disturbance, like the forest fire or the antibiotic use. But it doesn't always mean that they have to be completely eliminated in that community. And so in ecological succession, it requires the initial colonizers or the pioneering or primary species to alter the physical and that chemical environment, thereby allowing subsequent colonization by additional species. And then each additional colonizer continues to alter those physical and chemical properties, or the abiotic and biotic factors of that habitat, and therefore allowing subsequent colonization by more additional species. And this continues until there's a dynamic equilibrium reached in the community between the populations. So yeah, that would be that dynamic equilibrium is when we have everything reached, everything reaching equilibrium. That's what we're calling that climax community. So another example of ecological succession that's outside of the context of medicine is raw milk. We know that raw milk undergoes a really predictable 4-step process of succession of microorganisms during spoilage of milk. And so here we're looking at over time and the number of organisms, and then at the top we have acidity. So most acidic to least, or actually no, that's not how it's working. Right here we have the most acidic point, but they're measuring acidity at these time points. And so initially you have this growth of this Lactococcus lactis. It's a lactobacillus type of organism, and this produces acids in the milk. And so this acid production is then associated with the growth of this more acid tolerant species. So lactobacillus species are resistant to this really acidic environment, and so they then start to grow. And then after, that happens and the acidity of the milk is really high, that's why it's really sour when it's spoiling and you drink it. You then start to have these yeasts and molds that are able to grow. And so they take over and they start to degrade that lactic acid that's being there. And so by doing that, they're gradually decreasing the pH. So decreasing the acidity, raising the pH, decreasing the acidity. So it's becoming more basic. And then eventually you will have these bacteria that can degrade proteins that become active, which result in that really terrible smell and flavor that you get with spoiled milk. So it's a predictable process that we know, and it has to do with who's growing, then who can grow after that acid is produced. Any questions on any of this? All right. So let's talk about indicator species. So sometimes the presence, absence, or abundance of a specific species can reflect a specific trait or characteristic of the ecosystem. So we call that an indicator species. So basically, it's something that can be used as a proxy to indicate a change in the ecosystem. And so this can reflect the presence or absence of some environmental qualities or characteristics that are normally found in a specific place, such as like a unique microenvironment. So, for example, if an environment is supposed to be acidic and you assay it and you find that there's some organism that's growing in there, and that organism cannot grow in acid, then it's like, wait a minute, like you wouldn't be here if the environment is acidic, which is what it's supposed to be. So that's an indication that something has gone wrong. So that organism growing there basically is the indicator species telling you that something is wrong and that environment is not acidic like it should be because it can't grow. And so we use this technique or this phenomena of indicator species in water quality monitoring. And so basically, in order to test the water that you drink and you play in, recreational water for pathogens, it's really hard to screen the water for every pathogenic organism. And so basically we utilize this topic of indicators organisms to measure the quality of water. And coliform bacteria, I've mentioned that coli means colon, so generally they are bacteria that will come from the intestinal tract. So those are used as an indicator organism, and they basically signal for potential for disease. And so basically, in one way, and we're actually going to be doing this in lab tomorrow, one of the things we're going to be doing is testing the quality of different types of water sources and seeing if there's any potential for fecal contamination. And so one of the ways that we do that is we are looking for the growth of E. coli. So that's a key fecal coliform. It's something that is found in the gut, and basically when you poop, it's going to be found in your feces. And so if we find this bacteria in the water, then we won't know that there's potential for contamination, or if the water is contaminated, there's potential for pathogenesis in that water and it's not safe to drink. So there are different plate types that can be used to detect these things, and tomorrow we're going to be doing that. We're going to be using special plates called EMB plates that grow E. coli and specific organisms similar that will indicate that there is contamination in the water. So E. coli serves a really important ecological role here. So back to this ecosystem map, when we apply these concepts to the relationship between humans and microbes, we find that many of these same relationship types that work in important ways, and we really need to study them to understand them. So for humans and microbes, there are multiple types of relationships that are working at the same time, like I said last week. And so the mammalian host can be synergistic with the microbiota, it can be amensalistic, it can be a predator for the microbiota. So all these different relationships can exist at work at the same time. And that's because the host is a multicellular organism, and each cell in the human body has to undergo differentiation to become different types of cells. You have your immune cells, you have skin cells, you have cells that turn, into certain structural components like the epithelial cells, things like that. And so there's multiple different types of relationships between the microbes and us as the host. And so one of these things that allows this to be at play is that having really high diversity. So diversity nurtures resilience. Ecosystems with higher species diversity tend to be more resilient to things coming in and trying to disturb them. And so one of the ways that we study microbial communities and ecosystems is by measuring the diversity. So if the ecosystem has a diverse community of organisms, then they're not all going to be likely affected by a disturbance in the same way. So it's important to have that diversity so that you can have a resilient community. And the way that we measure diversity from an ecological perspective is by asking who is there and how many are there. And that's kind of split up into two questions. And so when we're asking who is there, we're measuring the richness, and richness can be low or high. And we're asking how many are there, we're measuring something called evenness, and that can also be low or high. So these are two sides of the same coin. And so there are many different metrics that have been developed that ask slightly different versions of these questions that help us really, understand who is there and how many are there. But it's important to know these terms, richness and evenness, and how they apply. So let's kind of go through a little bit of an example. And so here we're giving you a simple example of just some fruit. And so let's try to decide, is this low richness or high richness, low evenness, or high evenness? So in this first one right here, we have apples, pears, and oranges. So there are 3 different types. And so if you have just 3 different types, and we're asking the question of who is there, so we're measuring richness. Would this be low or high richness? Low, yeah. And then in terms of evenness meaning, how many of each one is there? Is it evenly distributed? Is there an equal amount, or is one kind of overpowering the community versus others? So would it be low or high evenness? Low evenness, yeah, good job. So low richness because there are 3 types of fruit only, and it's low evenness because there's a lot of this more common type, the apple, and there's fewer of this rare type. So let's try to do this one. Low richness or high richness for this one. Low richness, same thing. You have 3 types of fruit. And then what about evenness? High. So because there are a similar abundance of each type, that's a high evenness. And so this contributes to a more diverse community, which is good. All right, let's do the third one. Lower high richness. High richness, and then what about evenness? Exactly. So it's high richness because there's 7 types of fruit, but low evenness because there's a lot of this common type, few of the rare type. And the last one, high richness and high evenness. Good job, guys. You got this one. So yeah, same thing, 7 types of fruit, but there's a similar abundance of each one. There's two or three of each, and so that gives you higher evenness. Any questions on this? So this is all called alpha diversity because technically we're at like the first level of measuring diversity, and what that means is here we're talking about a like fruit bowl So just think of it in the terms of like a microbial community. If you're just measuring, what's in one person's microbial community, for looking at just one individual's gut, this is called alpha diversity. But there's two different types of species diversity. There's alpha diversity and beta diversity. So beta diversity is a comparison of the diversity between two different ecosystems. And so ecosystems can be two different humans, they can be two completely different, ecosystems in the environment. And so it's really measuring between people or ecosystems. And that's usually measured as the amount of species that change between the ecosystem 1 versus ecosystem 2, or person 1 and person 2. And the alpha diversity is just the diversity within the particular person or the particular area or habitat or ecosystem. Yeah.
So, since like your mouth and your gut are two different like ecosystems, could that also be a beta diversity?
Yes, absolutely. That's a really good point. Yep, that would be beta diversity. Yep, so if you were measuring just the diversity within your mouth, that would be one ecosystem, that would be alpha diversity. And if you were comparing between two sites in a human, that would be beta diversity comparison. Okay, and so, it's no surprise microbial diversity is really important for human health. There's a lot of evidence showing this now. And so over here, and this is, very well studied in healthy skin. These are all the different types of bacteria that occur on healthy skin, and you see there's a lot of diversity. And then when you have somebody that has atopic dermatitis or eczema or an inflamed skin, you'll see that there's less diversity of microbes. So usually, in the past, we've always thought that microbes are bad and they're germs, and having a lot is going to be bad for you. But this is where, the paradigm is shifting, and it's actually a diverse microbiota that leads to health. And so this is called the biodiversity hypothesis. And so, like I said, our whole entire microbial systems are connected within the human body. And so if you have a decline in this biodiversity, you're clearly depriving the community of certain microbes, which results in imbalance. And so in turn, this microbial imbalance will affect your immune response, because, we have our immune system reacting to things in our body, and changes in our microbes will influence our immune system. And all of this together then predisposes people to have a higher risk of inflammatory diseases when you have a decline in biodiversity. And this can be applied to really a lot of different types of diseases, not just inflammatory diseases. This applies to allergies, asthma, IBD, Crohn's disease. All of those are associated with a decreased biodiversity of the microbes in that area. So how do we study microbes and their environment in the lab? So there's obviously culture-based methods, which you guys have already been doing. We've been growing things in culture on plates, but there's also these culture-independent methods. And so the reason being because we really have only cultured or have been able to grow in the lab about 1% of the microbes on Earth. So there's a whole lot of other microbes that we have not been able to culture for various reasons, and so we have to use culture-independent methods, and so I'll explain what that means. So that's next. All right, let's just go through a little bit of media. So in order to culture bacteria, we need culture media, and that's just the nutrient solutions that are necessary for microbes to grow in the lab. And so there are two broad classes. There's others, there's a whole list of different classes of them, but in general, we need to give the bacteria the nutrients that they need to grow, whether it's liquid or solid media. And so a defined media means that you know the exact chemical composition of what is in the media. So we call that a defined media. It's defined, we know exactly what's in there, it's precise. We also use complex media, and so complex media is complex in the fact that it's composed of everything that any microbe would need to grow. It's composed of amino acids, different digests, yeasts, and different extracts and sugars and things like that. But we don't know exactly how much of each one there is. We just know if we kind of throw it all into the mix, then they're going to grow. So it's complex, but it's poorly defined. So it's important to be able to distinguish those. And so kind of going a little bit further there are some subtypes of media that can help you do certain things. So when you know the capability of your organism of interest, you can design a series of media to increase your chances of isolating it from a mixture. So nutrient-rich media is in the name. It's nutrient-rich, so it contains plentiful nutrients that will allow a variety of microbes to grow. And so in this case, we're kind of talking about having, a flask, say, with different types of microbes. And so if we put whatever is in that flask onto a plate that's nutrient-rich, more than one of those can grow, if not all, because it can enhance the growth of all of them. But then, you can also take advantage of something called selective media, which contains compounds that will selectively inhibit the growth of some microbes, but not others. So, for example, the media that we're using tomorrow, that EMB media that I told you about earlier, it is selective. Like I said, it's a way that we measure water quality because it can grow potentially pathogenic organisms like E. coli. And so that one is selective because E. coli is a gram-negative bacteria. And so that media has specific components in there that selectively inhibits the growth of gram-positive bacteria. So it's only selecting for gram-negative bacteria. And then there is differential media, which, hence the name, it contains something that allows you to differentiate between two things that can grow on the plate. So it usually contains an indicator. It's usually a dye or something that detects particular chemical reactions that occur during the growth of the organisms, and then allows a selection of the bacterial colony to grow a different way from that mixed population. So, so this right here is an EMB plate. And so they are known for this magenta color, and you'll see them tomorrow. And so this magenta color basically is that dye that is put into the EMB plate. So EMB plates, I said, were selective for gram-negative bacteria, but they are also differential. And so what that means is they can grow different types of gram-negative bacteria. There are gram-negative bacteria that are lactose fermenters that can, that can metabolize lactose, and there are some that are non-lactose fermenters. And so basically this dye that is in here is the indicator, and it can then be used by lactose fermenting bacteria so that when they grow on these plates, they grow in this really cool, like golden greenish color. And then anything else that's non-lactose fermenting can't pick up this dye, and so they grow regularly. So they can both grow. They're both gram-negative because they're selected for, but you can differentiate them based on one of them having the ability to ferment lactose and picking up that dye and changing its color as it grows. Does that make sense? So media can be selective and not differential, or it can be selective and differential, or it could be not selective but differential, so it's important to recognize those differences. And so sometimes you can introduce nutrients or environmental conditions in the media that only allow the growth of an organism of interest, and usually it's because you're enriching it or you're mimicking its natural environment. And so basically what this means is that you know exactly what an organism needs to grow, and you try to mimic that in a lab and just enrich for that one organism. And so by doing this, if it grows, you're technically proving the presence of that organism, but it doesn't necessarily prove that an organism doesn't inhabit an environment. And so being able to isolate an organism from environment doesn't say anything about its ecological significance. This is just straightforward. I'm trying to enrich the growth of this. If it grows, I know that it's there and that's it. You can't really tell anything more about that, but it is a way for us to try to isolate certain things from different environments. An example of this is if you take some soil and you put it in this media, there's a lot of different types of bacteria in that soil that are going to be trying to grow, and they probably can grow, but there are these certain microbes that are nitrogen fixing bacteria, so we kind of talked a little bit about that. They're nitrogen fixers, and so let's say this media here is enriched for the growth of those nitrogen fixing bacteria. So basically, those ones will start to grow up and outcompete the others because the media is selecting for just those and not the others that are in the soil. Any questions here? Okay. And so whether it is liquid or solid media, in order to successfully cultivate a microbe, it's important to know the nutritional requirements and be able to supply them in the proper form and proportions in a culture media. So this is actually a really big downside of culturing organisms because you can know what it needs, but you might not know exactly the proper form that it would be growing in its natural environment or the exact proportions of how much of everything you need. So that makes it difficult. So in order to successfully cultivate or isolate microbes, you have to know the nutritional requirements, what it needs, the form that it's in, and the proportions of that culture media. And so culturing microbes can be difficult. It's no secret, but microbes that are cultured in the lab are frequently only minor components of the microbial ecosystem. So, if you think about it, if you're trying to study something, I've said this before, and you're trying to study it in a lab setting, you have to really think about the fact that where it's coming from is this whole entire ecosystem where there's other organisms there as well that are going to be, either have some relationship with the organism you're trying to study. So if you're trying to, for instance, if I'm trying to study, a certain microbe in the lab that comes from the gut, and I just grow that one up by itself, I'm being, absent-minded in the fact that I'm not thinking that, in the gut actually it's growing with all of these other microbes and they have either beneficial or negative interactions with each other, and you have to account for that in the lab as well. And so some microbes are quite challenging to culture, and there are many reasons for this. You can either have not the correct nutrients for the culture, you can just not have enough of a limiting nutrient or too much limiting nutrients, and so you can also have the wrong pH, oxygen concentration, temperature, and then you could also be missing those syntrophic partners that I was talking about, and then sometimes you can get kind of sick of it and throw it away because nothing's growing, but it really needed 2 more days to grow, and so your microbes that are missing are just too slow to grow. So it's really an exercise in patience as well. Okay, so the problem in studying microbial communities from any site. And so the majority of the bacteria won't grow in nutrient media in the lab. And so this idea was officially named in the 1950s. This is called the Great Plate Anomaly. So basically what happens here is if you take a sample from any environment, whether it's an ecosystem like a forest or the gut or anything from any environment, and so you basically take that sample, you mix it with water, you vortex it and do your thing, and then you're trying to culture it, and you try to put it on a petri dish, and then you take that same one and then you also put it under a microscope to try to count the number of cells, and so you'll see that the numbers, you'll see that the number of colonies that end up growing on the petri dish are going to be substantially less than what you see on the microscope, and so this is one of the most profound puzzles in microbiology, and so basically there are many efforts to try to grow that permaculture media that haven't been successful. And so essentially what this is saying is that, you know, if you take something and you're trying to count how many cells there are, you'll see more under the microscope, but when you try to put it in media, in something that it's supposed to like, you'll see less of it. So it's kind of really puzzling there. And different molecular methods have suggested that only a very small minority of bacterial species have been cultured from all environments. There's really about like 1%. So how do we get around this? How do we study the 99% of bacteria that exist, but we can't grow in a lab yet? So everything, including microbes, has DNA. So DNA is the answer to this. Basically, all microbes have it. It's specific for different organisms. And so basically, if we know the DNA, you know, the central dogma of life is DNA to RNA to protein. So if we know the DNA, then we know the RNA. And if we know the RNA, then we can predict the proteins. And proteins have functions. So DNA can really help us in terms of understanding the functional capabilities of microbes as well. So it's not just the growth. And so DNA sequencing has really changed the game here. So the method I'm just going to briefly talk about is the Sanger dideoxy method. And so basically, DNA is the main information carrying molecule. And so Fred Sanger in the 1970s, he won the Nobel Prize for this. He won 2 Nobel Prizes actually. But basically, he used this the DNTPs, or deoxyribose nucleotide triphosphates, to figure out the sequence of DNA. And so I am going to have this lady explain this a little bit better. Well, it worked yesterday. Okay, well, if it doesn't work, then you don't need to know it. But just to give you a little bit of, you know, a summary of what this is basically this guy, he screwed with the DNA sequences, the A's, C's, T's, and G's to like terminate them. He took the hydroxyl group at the end of each one of those, and he basically will terminate them. And so by doing that, he like ran them through a gel matrix. So if you guys have heard of gel electrophoresis, basically it's, you have this buffer, this like liquid buffer, and DNA is negatively charged. So you have these electrodes where there's a negative charge where you put the DNA, and then a positive charge on the other end. And so basically the DNA will run towards the positive charge because it's negatively charged. And based on the size of that DNA, it's gonna basically stop at a certain point. And so then you can measure basically how big or small it is based on that. So you're separating them by size. And so basically by doing that, so the smallest fragments are going to finish first, and because they are smaller, so they're less heavy, and so they're going to kind of stop and move through quickly. And so then he put it through this other detector, basically that has colors, and the sequence of the DNA will ping a color, and that let him know whether it was an A, C, T, or G. And so that's just a method to use. You guys don't have to know all these details. But yeah, essentially, it's been really, really used. It's still used today. It's a very accurate method. You basically will take the DNA from something. You'll isolate the DNA in the laboratory, and you can submit it for sequencing. And ultimately, what you will get is the entire code, the A's, C's, T's, and G's from beginning to end. And so once you retrieve that, we have a database, we call it BLAST, and so basically once you have all that file with all the ACs, T's, and G's of whatever it is, that unknown that you sequenced, you can put that file into this database, and then it will match up to something that's already in the database, and you can figure out what it is that you just sequenced. So that's a little spiel of Sanger sequencing. So basically it's using these fluorescently labeled nucleotides, and then you're able to match up your sequence to something that's in the database to know what it is. And the other huge scientific innovation that revolutionized microbiology is the invention of PCR, or polymerase chain reaction, in the 80s. So this is a key technique that was used in microbiology that gives us the ability to selectively pick and copy certain regions of DNA from any organism. So let's see if this one will work. I guess it's not going to work either. Okay, but I can explain this one. So basically, PCR is having DNA replication in a test tube. So what we normally do is we know, say I have an organism and I want to basically try to, let me give you an example here. So you have some organism and you want to try to, know like what it is. And so what you'll do is, if you think that it could be a certain, bacteria, for instance, and you know that this bacteria makes a certain gene, then what you can do is put that DNA in a test tube, and then you'll create these things we call primers that align to that gene of interest in that DNA sequence. And so those primers will be complementary to the DNA. So you'll put those also in the test tube, and then you'll add a DNA polymerase that will get that reaction going. And you'll often have something called a Taq polymerase. That's the DNA polymerase. And then once you put those things together in the test tube, you run it through a PCR reaction where there's a lot of heating and cooling and repeating of the cycles. Basically, the heating step will separate the DNA strands because heating will denature things. And so it will separate them, and then it will let the primer know, like, okay, it's separated, and the primer can then go to the specific spot on the DNA region where it's complementary. And then you'll have the polymerase starting to make, copies of that. And then when you cool, when the cycle knows to cool itself, it knows to then anneal those primers until you get a copy of that gene. And then, and so basically that cycle repeats over and over and over again. So you get many, many different copies of that gene of interest in that bacteria. And then you can run it on a gel and actually see the size of it, and if it matches that gene of interest, the size of that. So that's another way to identify things. Apologize if those did not work. I will try to extract them and put them up on Blackboard for your reference. But that is what I have today for this. I do have worksheet number 5 over here, so pick one up. It's going to be due next week. Go through these. These are from lectures 11 and 12. They should help you with reiterating some of the concepts. And then next week, we'll talk a little bit more about human microbiome stuff.